Greetings and welcome to a series where we discuss how the scientific definition of reality and of ourselves could suddenly change in the very near future. There is strong evidence that our reality could be structured holographically, and that you and I could be structured holographically. In this episode, I will show a clearer picture of what it means to be holographic by investigating the holographic nature of biology. In my previous video, we discussed how a source of laser light, a diverging lens, and a specially prepared piece of film could produce a three-dimensional hologram. In this case, horses pulling a gladiator in a chariot. One of the defining features of a hologram is that if you cut the film into smaller pieces, each piece will contain the entire image of the hologram. That image will be the same size as the original, although its resolution will be lower. Holograms can also be projected onto a screen as a two-dimensional image. Watch now as the technician punches out a tiny fragment of film, only a few millimeters in diameter. He then holds up the tiny fragment with a pair of forceps and allows the laser light to shine through the fragment and onto the screen. The image produced by the tiny fragment is the same size as the original. So a hologram of a person can be viewed as a three-dimensional image like this, or projected onto a screen as a two-dimensional image like this. This is useful for demonstrating that any tiny fragment of the hologram contains a full-size version of the image, though at a lower resolution. In this sense, a hologram of a human being has something in common with a real human being. The body of a human being contains about 37 trillion cells, which are generally too small to be seen with the naked eye. Each cell in the body of person A contains the blueprint of the entire body of person A, and a copy or clone of person A can theoretically be grown from one of these tiny cells. Although this is not currently permitted for moral and ethical reasons, cloning has been successfully demonstrated in animals, such as these sheep. So we could say that in both holograms and biological life in general, the macrocosm contains the microcosm, and the microcosm contains the macrocosm. This phrase encapsulates what it means to be holographic, and it is often used in channeled information to convey the holographic nature of the universe. Now let's take a look at the development of a salamander from a single cell. After fertilization, the single cell divides into two cells. These two cells then divide into 4 cells. The 4 cells divide into 8 cells, and so on. The cells continue to divide and grow for a period of about 2 to 4 weeks, ultimately leading to the formation of a fully formed and hatched salamander larva. Although the production of life is one of the most common occurrences on our planet, how it all began is said to be biology's biggest mystery. How did a collection of lifeless atoms and molecules evolve to form the diverse biological life on our planet? There seems to be no obvious explanation as to how this could have occurred naturally. In fact, scientists at Oxford University studied the prevailing theories of evolutionary biology and concluded that we find a substantial probability of there being no other intelligent life in our observable universe. One news website summed up the study in lay language like this: "We're as good as it gets. Intelligent life is extremely unlikely to exist anywhere else in the universe because it took a series of miracles for humans to evolve." The word miracles here is used to characterize outcomes that are statistically extremely unlikely, so unlikely that they probably haven't occurred anywhere else in the universe. However, some scientists believe that there is a more natural explanation for the development of life. Biological structures could have originated from sound or vibration. Let's take a look at the field of cymatics, which is basically sound made visible. This is a body of water in a small dish attached to a loudspeaker. The speaker is vibrating at a constant frequency, producing waves in the water. These waves form specific interference patterns at specific frequencies. The light shining on the water is also producing an interference pattern, and here we see that pattern shown as it looks from directly above. Note that the words sound and vibration can be used interchangeably in this context. Here is a more sophisticated instrument for producing cymatic imaging, called the cymoscope, and here is a demonstration of how it makes a piece of music visible. John Stuart Reid, inventor of the cymoscope, shows a potential connection between cymatics and the origin of life.
Here are some slides to show you how I got into all of this in the first place in the laboratory. Almost every day in my general work, looking at different frequencies for different reasons, different projects that I'm working with, I come across images like this that are showing what look like early life forms. I've got hundreds of them. There was just a few just to show you. But, you know, that makes me think, okay, is there some connection between sound and life? Well, I think that very much that there is. And there's something showing the cross-section through DNA, tenfold, and there's a tenfold Faraday wave model.
Biologist Rupert Sheldrake also believes these vibratory patterns are the origin of life.
This is an eightfold pattern, and by vibrating at different frequencies, you get a series of patterns: sixfold, eightfold, tenfold, 12-fold, 14, 16, 18, 20, and it goes up further. So there are these symmetry patterns, and these are, as it were, resonant frequencies. One of the people who's done most research on this in relation to biological forms is Alexander Lauterwasser here in Germany. And what he's shown is that these radially symmetrical patterns that you get vibrating water have distinct analogues in biological forms like flowers. And they also have analogues in radially symmetrical structures. These are pollen grains. Each of these are from different species, and each species has a particular pattern of the pollen grain. These are single cells. And why this is important is that current thinking about biological morphogenesis is heavily molecular, and most biologists think it all happens just by switching on and off genes. But in a single cell, switching on and off genes can't explain the morphogenesis of a single cell, because the genes are the same for the whole cell, and what's switched on and off is the same for the whole cell. So how does a cell, a single cell, create complex form? Well, I think the only answer is through a morphogenetic field. And instead of morphogenetic fields being conceived of as they currently are, primarily in terms of diffusing chemicals, I think they're actually vibratory patterns.
As we watch the development of a salamander in its egg, it surely seems logical that this is a reenactment of how the salamander evolved in the first place. When this very watery single cell divides into two identical cells, it could be undergoing the same type of process that takes this dish of water from this pattern to this pattern when the vibrational frequency is increased from 9 to 11 Hz. We can clearly see that the new frequency has divided the pattern into two sets of point-centric waves. So now we have a mechanism which produces the fundamental building blocks of a hologram in which each set of point-centric waves represents a cell. Each cell has universal extent, and therefore there is universal entanglement. As the number of sets of point-centric waves increases, so the number of cells increases. Each cell is a holographic cell, since it contains the pattern of the whole embedded in it, due to the universal extent of the waves. In other words, the microcosm contains the macrocosm. the complexity of this organism could increase over millions of years as the natural processes of biological evolution come into play. But all throughout, the holographic principle that the macrocosm contains the microcosm, and the microcosm contains the macrocosm, would be maintained in the cells and the DNA of the salamander. Now that we have a clearer picture of this principle, we can clearly see that biology is holographic. But how did it get to be holographic? The mainstream theory of chemical evolution proposes that atoms and molecules combined in a primordial soup of chemicals. These chains of molecules became increasingly complex until they ultimately began the process of cell division and reproduction, which is holographic. This is the evolutionary step that scientists find the most difficult to fathom. So, is biology holographic due to an extreme statistical anomaly, or is biology holographic because the underlying physics of the universe is holographic? If the universe is indeed holographic, then this holographic principle must apply to every level of the universe. A galaxy must be a microcosm of the universe, and so must a solar system, a planet, a human being, a biological cell, and an atom. They must all have the same inner workings as the universe itself, including biology. But what is this universal infrastructure that all these levels have in common? That will be the topic of discussion in the next episode.